So we continue with the discussion of the heteronuclear correlations. As I mentioned, the most common experiment here is the heteronuclear single quantum coherence, which is also called as HSQC. Okay. Now how does this experiment go? Just look at the pulse sequence. Of course there are two channels here. Now you apply the pulses on one channel, these are all the it is called as the proton channel. You selectively apply pulses here on the proton, these are not selective pulses, these are non-selective pulses, these are hard pulses. Then you can also apply pulses on the carbon channel. So we have the carbon here. So you require two different coils in your spectrometer. You have a carbon coil and a proton coil. So pulses on proton coil are applied with using the proton coil in this channel, and pulses to the carbon 13 are applied using this channel here. And there will be no interference between these two because these are so widely different frequencies. If this is 500 megahertz, this is 125 megahertz. If it is 600 megahertz, this is 150 megahertz because 1/4. And if it were nitrogen 15 here, this is 1/10 of the proton. If it is 500 here, it is 50 there for nitrogen, and like that. Okay. So now how does this experiment work? So here we start with the proton. We start with the proton magnetization, that is we represent it as Hz. And then this sequence is already what we have seen before, this is called as the inept sequence. So you have proton magnetization 90 tau, 180 tau, and that there is a 180 on the X channel as well here. Okay, let me write that as 190 and 180. We write it as 180 and this is also 90. Okay. So now when I do this, so this is called as the inept experiment. So during this period the proton is now transferred from the Z magnetization, it comes to the transverse magnetization, and this will evolve during this period with the coupling to the X nucleus, carbon 13 proton coupling, and from here these two 90-degree pulses, the magnetization is transferred to the carbon. So this is the inept sequence. Okay. Let me write here, this portion is your inept. Inept transfer. Okay. So therefore the magnetization flow, how does the magnetization flow? It starts here, comes here, and then it moves to this. Now this is, now the magnetization here is on carbon. How do we know? That is what is represented by 2HZCY. So this CY means it is the carbon magnetization here because it is on the carbon 13. So the carbon magnetization which is transverse, this will evolve during the period T1, and in the middle of the T1 period I apply a 180-degree pulse onto the proton channel. Okay. So when I apply 180 pulse to the proton channel, what does it do? It actually decouples proton carbon coupling. So this is the spin echo period from here to here, T1 half, 180, T1 half, this is the kind of a spin echo period, and during this period there is proton carbon decoupling. So we can write here proton carbon decoupling. That happens, and the carbon frequencies will evolve with the carbon frequencies. What frequencies will be present here? Let us say the frequencies which are present, I will have it as omega c. Carbon frequencies are present, whatever carbon is attached to the proton, and that from whichever proton magnetization is coming to the particular carbon, and that is what is evolving with the frequency of carbon. So this will continue like this, carbon magnetization evolves, then it is transferred back to the proton. Magnetization goes back to the proton by these two 90-degree pulses. This is 90 here, this is 90 here. By these two 90-degree pulses, magnetization goes back to proton. This is again an inept. Okay. Now here it is a reverse inept. This is the reverse inept transfer, this is the refocusing. We have got the magnetization on the proton once more, but this is 2HYCZ. So this is antiphase proton magnetization, and here it was the carbon magnetization which is antiphase with respect to the proton, that remains like that only. When I apply the 290 degree pulses, it is converted to antiphase proton magnetization, and then during the refocusing period I will get here the in phase proton magnetization. So Hx represents in phase proton magnetization. So the in phase meaning then when I detect the signal during the period T2, I can decouple carbon. All the proton carbon coupling is removed during the T2 period. Okay? Alright. So what we have now? Only the proton frequencies are present during the T2 period, there is no coupling. During the T1 period what do I have? I have only the carbon frequencies, I do not have the proton frequencies. Okay. So let me repeat this here. We start with the proton magnetization which is the Z magnetization, and during the first inept sequence we transfer the magnetization to the carbon, it comes as antiphase magnetization. Now what does this tau 1 depend upon? Tau 1 depends upon the time required for this inept transfer. Typically in the case of proton carbon, this will be typically tau 1 will be of the order of 1.7 millisecond, that is approximately 1 by 4j. Tau 1 is equal to 1 by 4j Ch. So if let us say your proton carbon these are one bond couplings. So the one bond coupling, if it is let us say 150 hertz, assume 100 is in the higher side, if it is 150 hertz, so 1 by 4j is approximately 1 by 600, therefore approximately 1.7 milliseconds. So if JCH is equal to 150 hertz, one bond coupling, so tau 1 will be 1 by 600, and that will be approximately 1.7 milliseconds. So therefore it is extremely small period, very small period you apply this, and then you have the 180 degree pulse and you have the transfer coming here. The magnetization remains as 2HZCY, and then it goes back to, after frequency labeling with the frequency of the carbon during the T1 period, it goes back to the proton, and this refocusing period also tau 2 should be roughly equal to T1. Tau 2 is, it need not be exactly equal, there are other advantages of having T2 not equal to T1, but by and large in the simplest case tau 2 can be equal to simplest case tau 2 can be equal to tau 1. That is the refocusing, so you get Hx there, and you have then the decoupling happening from the on the carbon channel. So proton carbon coupling is removed during the T2 period. So therefore you have in the T2 period only the proton frequencies, and the T1 period we have only the carbon frequencies. Okay. Now let us see what happens, how does the spectrum look? See, this is what it is. Along the F1 as I said, or the omega 1, the T1 evolution I have only the carbon frequencies. Along the F2 I have only proton frequencies. Therefore this is like cross peaks only, right? The entire spectrum has only cross peaks, there is nothing as a diagonal here. We have only cross peaks, you have the proton carbon correlation spectrum. So if there are 4 different carbons here, you will have single peak, one peak each for this. Now notice the carbon frequency range is quite high, almost like 100 ppm you can have, forget the carbonyl, and if you take the aromatics it can go up to 120, 130 ppm. There is a whole range, a wide range of frequencies here, and the proton frequencies can go to from the all the way from 0 ppm all the way to 10 ppm. So you will have all of these proton frequencies here. So one peak each for one carbon. So this is very, very simple, quite a substantial enhancement in the information content. Now here I have shown you the schematic with regard to the carbon, but you can also have the nitrogen here, and typically in the case of proteins we do proton N15 correlations. So for proteins we have proton nitrogen 15 correlations. These are all one bond couplings. every amide group of an amino acid residue produces one correlation peak, okay. So, one peak per residue. So, this is the, and this produces one peak per residue. Along the backbone, I am looking at the backbone, so it will produce one peak per residue. Now this is an experimental spectrum of a particular protein. So it does not matter which protein it is all labeled there. You can see here this is the N15 axis, the F1 axis is the N15 axis, that is indicated here, goes from 120 ppm here, okay, 110 ppm to 140 ppm almost, about 30 ppm range, and here you see such a wide distribution of peaks, and these are all correlation peaks only, nothing else. And on the top you have the glycines, then you have the threonines here, then you have various other ones, asparagines, alanines and things like that. This is the little crowded region. By and large this area is more crowded. This contains all the aliphatic amino acid residues, the leucines, okay, and the alanines, isoleucines, glutamine, everything will come here, all the aliphatic ones. And this is the blow up here, you can see this portion is blown up here in this the right side spectrum. All of them are very distinctly seen, and you can actually identify all of these peaks. This is the spectrum of a something like about 130 amino acids, and in a proton spectrum it is impossible to get this kind of a separation. Impossible to get a certain kind of separation, proton correlation spectrum. And in the N15 it is easy to get. The N15 lines are also quite sharp, therefore you can resolve these ones very well. And since there is only one peak per residue, okay, one peak per residue, you can simply count the number of peaks here and see whether you have got all the peaks or not. So if you have a protein of 100 residues, you should get 100 peaks, barring prolines. The prolines do not have the amide proton, so the prolines will not produce a peak here. All other residues will produce a protein, produce a peak. Therefore by simply count the peaks here and see whether you have got the proper spectrum. Therefore this experiment is called as the fingerprint of the primary sequence. Of the primary structure. Essentially it is a amino acid sequence. This is which one is we are talking about now? Proton N15. Proton N15 correlation spectrum is a fingerprint of the primary structure. This is I here, it looks like a J. So this is I, fingerprint of the primary structure. So this is an extremely useful experiment to perform. Okay. Now can we do something more than this? Okay, we got the HSQC spectrum, we got the NOESY spectrum, 2D correlation spectrum. Now we can we combine these two informations? Well, you can do that. So these are called as, then you can combine HSQC with TOXY or NOESY. The pulse sequence can be designed approximately that way, and what are you going to get here? The HSQC spectrum, let us say we are talking about the N15 proton. So let us say I have here N15 and I have here the proton. Assuming that I have one peak here which belongs to a valine residue. Let us consider a valine residue. So what is a valine residue? NH, okay, CH, okay, this is the C alpha H here, then I have the C beta H, then I have 2 methyls. These are the gamma 1s and the gamma 2s. In this case what will I see? I will see only one peak here in the HSQC. in the HSQC. But now what I want to do is I want to transfer the information. What we have transferred here, we initially started with the proton, transferred to the nitrogen and back to the proton to measure it. Now what I want to do is once it comes back to the proton, I want to transfer this to the alpha proton, to the beta protons and the gamma protons. I want to transfer. This I can do by the TOXI or the NOESY. Okay. So therefore what will I do? I combine this with either the NOESY or the TOXI. This is the proton axis here and this is my NH proton, and here I have the C alpha H, I have the C beta H, and here I have the 2 methyls. I have this peak here, this is my HSQC spectrum, NH to the N15, on this axis I have the N15. But now I want to relay this information, once it is on the proton I will relay this to the other protons. So what will I get? I will get a peak here, I will get a peak here and I will get peaks here. Okay. So therefore this is my TOXI HSQC. So I am using the full proton range here, while I will have only the correlation peak from the proton to the nitrogen 15. By combining this with the TOXI mixing sequence or a NOE mixing sequence, I can generate this information from NH proton to the alpha proton or to the beta proton or to the gamma protons. And if I have another residue, some other residue, let us say, so let us say I have only the glycine, suppose I have a glycine, this is for the valine, this was for the valine case, okay, this was the valine case. Suppose I have the glycine, now let me use a different color for that. Suppose I have a glycine here, which is usually on the top of the HSQC spectrum, okay, and this is at a different NH chemical shift, and let us say now that NH chemical shift is here, I will have this peak here, and then I will have to the alpha protons of the glycine. If the glycine has 2 alpha protons, if the 2 are non-equivalent, then I will see 2 peaks here. Okay, now this will be for the glycine. So by doing this you can actually identify all the peaks of the amino acid residues, individual spin systems as well, from a combination of these sequences. Okay. Now if you do a TOXI, this will happen within the transfer within the same amino acid residue. But if I do a NOESY, this also will have sequential and long range correlations. See here there is no diagonal, you will only have cross peaks. Therefore in either case, if you combine this making use of the dispersion of the N15 chemical shifts, then you will be able to separate out and identify the individual amino acid residues by looking at these relays. Okay. It has also enormous structural information. Okay. So in the next slide I will simply give you the pulse sequence how it is done. We will not go into the details of that one there, but I will just indicate to you the pulse sequences. Okay. So here I have the HSQC TOXI. So it starts in the same manner. I have 2 channels here. Let us say I call this as N15 and this is 180 degree pulses on both the channels. This is the proton channel, this is tau 1, tau 1, then I have the 90 degree here, the 90 degree here, this is nitrogen 15 and this is T1 by 2, then I have a 180 degree pulse here, no pulse here, then this is again another T1 by 2, then I have two 90 degree pulses as in the HSQC, and after this I have this tau 2, this is okay. Now these are all 90, 180, 90, 180, this is 90, this is 180, okay, and this is 180 here, this is 90, then this is 90, this is 180, and then I have another tau 2. So then I come to this point here, this is the refocused point, okay. This is again tau. Here I introduce what we call as the toxi mixing. Toxi mixing is used, and after that I collect the FID, and when I collect the FID here, then there is broadband decoupling. So this portion is HSQC from here to here, and then I have combined that with the toxi. So this is the HSQC toxi spectrum. I can do the same thing for the NOESY, HSQC NOESY. Let me use the different color for that. So this portion is the same. Okay, and then I have again this also is the same here. Now at the end of this what we will do is the ones different, the NOESY mixing I have to introduce. For the NOESY mixing I will put here a 90 degree pulse which creates a Z magnetization, and from here onwards it is a mixing time, there is a tau m, this is the NOESY. And at the end of this I have another 90 and we collect the FID, and when we do that then we decouple this. This is broadband decoupling. So therefore it goes exactly the same way, except when we want to do the NOESY mixing, then you will have to use this sort of a modification in your pulse sequence, and you have depending upon, you can adjust your tau and do it in the same way as you do in the normal 2D experiment in the normal NOESY, or simply in the one dimensional NOEs. This depends upon the T1 period. The same conditions, same criteria are applicable here as well. So this is proton and this is nitrogen 15, and you get the relay information in the NOESY as well, and also the sequential connections. Okay. So this is the 2-dimensional experiments. Now let us see, we can go forward in the little bit more complicated situation. Okay, here we extend it. Now how do we use this, combine this to improve the experimental resolutions? Okay. Now we combine two kinds of 2D spectra and generate a 3-dimensional experimental sequence. A 3-dimensional experiment, how is it produced? A 3-dimensional experiment is produced by doing a 3D type of time data collection. So 3D experiments. So this is typically, as with the schematic, you have a period here, the T1, then you have a mixing, this is called as M1, then you have the T2, then you have a mixing which you call as M2, then you have the data collection which is T3. Yes. So here we will generate one frequency axis F1, F2 and F3. So we can have different combinations, okay. So M1 is a mixing which will produce one kind of a correlation, M2 is mixing which can be another kind of a correlation, and these can be heteronuclear experiments as well, the combination of heteronuclear experiments as well. I will illustrate this to you how this is going to be useful in 3D or 4D. If you want to go to the 4D, then of course you must introduce one more step there as mixing sequence M3 and then the T3. When I say M1 or M2, it includes all of that what I described before. For the HSQC, what sort of a mixing you use, for the Toxi what mixing you use, for the NOESY what mixing you use. And this is the benefit of this is schematically indicated in this slide. Let us see here, you have a 2D proton-proton spectrum here, proton-proton spectrum, and at this point you have so many peaks here. The one particular amide proton is producing those so many peaks at the same NH chemical shift, it is producing so many peaks. It is difficult to figure out where are they coming from. Now if I do on the same sample a proton nitrogen 15 correlation spectrum, this is the HSQC spectrum as I indicated to you, then you will see at the same NH proton I am seeing 3 peaks here, 3 N15s. 3 N15s, what does that mean? It means at least there are 3 amino acid residues who have the same NH chemical shift. 3 amino acid residues which have the same NH chemical shift, and those ones are showing me these 3 nitrogen 15 peaks, because the nitrogen 15 chemical shifts, both those 3 amino acid residues are different. Now what I do is I take this spectrum, pull this apart using the N15 chemical shift in a 3D manner. So I put the N15 chemical shift on the third axis, and these two axes are proton-proton axis. On the third axis I put the N15 chemical shift. In other words, all these peaks which I am having, the 10 peaks which are present here, I am pulling these along the nitrogen 15 axis. So what I get? I get here 3 peaks, which is easy to understand now. Well, this can be NH to the alpha, to the beta and to the gamma of a particular amino acid residue, and then another amino acid residue. This produces 4 peaks, which is certainly possible. You have from the NH proton to the alpha, to the beta, to the gamma and the delta of that amino acid residue, and you have the third N15, third amino acid residue whose N15 chemical shift is here, that is this. So we have this N15 chemical shift is this one here, this N15 chemical shift is this one here, and this N15 chemical shift is this one here along the N15 chemical. I have here 3 peaks. This may well be the NH to the alpha and then to the 2 betas, maybe the threonine or the serines and things like that. So you have these 3 ones here. Now if there is any further ambiguity in these ones, you can also use carbon chemical shift. Then you go to the 4D. We will not go into that detail here. So you can use the carbon chemical shift to separate these ones out further on the basis of the carbon chemical shifts. So then you will have all of these, each plane is now separated into multiple planes here having different carbon chemical shifts here. This becomes a 4D experiment. So you have the 2D experiment, proton-proton, and you combine that with a nitrogen 15 chemical shift, you generated a 3D spectrum wherein the proton-proton correlation peaks are separated out on the N15 axis, and then if you have further complications arising, which may not be, maybe may not be, if it happens then of course you need the 4D. But of course all of this will increase your experimental time as well, because the T1, T2, T3 all of them will have to be independently incremented, so that will take quite a toll on the experimental time, on the machine time. So by and large one tries to minimize these number of dimensions, you try to get as much information as possible from the 2D or the 3D experiment. Okay. So therefore this is going to be extremely useful in all our future experiments. So this is an experimental spectrum of a 3D spectrum. See here you have proton chemical shifts on one axis, the nitrogen chemical shifts on one axis, and the C13 chemical shifts on the third axis. All these are different chemical shifts. So the peaks are appearing in now in a box, okay, and if you take projections of this onto these individual planes, then if you take a projection on this bottom plane, then you get nitrogen 15 C13 correlations. Nitrogen 15 C13 correlations you will get. If you take a projection on this plane, then you will get the nitrogen 15 proton correlations, okay. So therefore depending upon which projection you take, you can get different kinds of correlations there, and so therefore this is how the 3D spectrum look like. Now you see, the instead of in a plane, now you are putting the peaks in a box. This is enormous visualization possible. If you go to 4D, it is not easy to visualize this, but up to the 3D you can visualize this. Okay, so I think we will stop here.